Good afternoon, all. In today's lecture, we are going to discuss about the hybridization methods in the self-pollinated crops. Okay, in the last lecture, we have discussed about the field technique which is used in the hybridization. Also, we have discussed about the advantages and the limitations of the hybridization method. Okay, hybridization, it is nothing but the crossing or the mating of the two plants which are having the dissimilar genotypic constitution. Okay, hybridization is nothing but the crossing or the mating of the two plants which are having completely different genetic constitution, or the plants which differ in their genetic constitution. Okay, that process we call it as a hybridization. Okay, on field, the hybridization process, it is accomplished into the 7 steps. Okay, the process, it is accomplished in the 7 steps. The first step is the choice of the parent or the selection of the parent. Then second one is the selection of the parent. The second one is the evaluation of the selected parent. Then the third one is the emasculation. Fourth one, bagging. Fifth one, tagging. Sixth one, pollination. And the last step is the harvesting and the storing of the F1 generation seeds. Okay, that is how the hybridization it is achieved on the field in the 7 steps. Okay, it involves the two most important, two critical technical operations: emasculation and the pollination. Okay, we have discussed the different methods of the emasculation and the pollination in the last lecture. Okay, in the theory examination, the question may be asked, long answer question may be asked for the 20 mark as what is mean by hybridization, and give a detailed account on the hybridization technique. Okay, I'm repeating this one. In the examination, the long answer question may be asked for the 20 mark as what is mean by hybridization, and give a detailed account on the hybridization technique. Okay, today we are going to discuss the hybridization methods in the self-pollinated crops. Okay, because the handling of the F1 generation and further segregating generations F2, F3, F4, or F5 generations, this is completely different for the self-pollinated crop and the cross-pollinated crop. Okay, methods of the handling of F1 generation and the further subsequent generation are completely different for the self-pollinated crop and the cross-pollinated crops. Okay, handling of F1 generation. In the last lecture we have discussed up till the harvesting and the storing of the F1 generation seeds. Okay, but how the F1 generation and the further segregating generations are handled, that is, these methods are completely different for the self pollinated crop and the cross pollinated crop. Okay, and in today's lecture, first of all, we are going to discuss about the hybridization methods in the self pollinated crop. Okay, okay, here you can see the handling of F1 generation. Handling of F1 generation is different for the self pollinated and the cross pollinated crop, and that is we referred it as a hybridization methods. Okay, the in today's lecture we are going to discuss the hybridization methods in the self pollinated crops. Okay, the hybridization methods in the self-pollinated crop, there are 4 methods which are used for handling F1 and further segregating generations. Okay, these 4 methods are the pedigree method. First one is the pedigree method, second one is the bulk method, third one is the backcross method, and the fourth one is the multiple or the composite cross. Okay, for handling the F1 and the further segregating generations in the self pollinated crops, four methods are used. The first one is the pedigree method, second one is the bulk method, third one is the backcross method, and the fourth one is the multiple or the composite cross. Okay, in today's lecture we are going to discuss the two methods. The first one is the pedigree method and the bulk method. Okay, and tomorrow we will discuss the backcross method and the multiple or the composite cross. Okay, today we are discussing about the two methods which are used for the handling f1 and further subsequent generations in the self pollinated crop. The first method is the pedigree method and the second method is the bulk method. Okay, every selection method, it may be pedigree method or the bulk method or the backcross method, or it may be the multiple or the composite cross. Okay, every selection method has its application under specific conditions. Okay, every selection method has its application under specific conditions, and these methods are used for the definite advantage. To get the definite advantage, these methods are used. Okay, each and every selection method has its application under the specific conditions, and the methods these are used to get the definite advantage. Okay, in this one, the first method is the pedigree method. Okay, the first method here we are going to discuss is the pedigree method. Okay, here what is mean by pedigree? Pedigree means it is nothing but the record of ancestry of an individual selected plant for various generations. Okay, pedigree means, here is the definition of pedigree: a record of ancestry of an individual selected plant for various generations is known as the pedigree. Okay, record of the ancestry of an individual selected plant for various generations is known as the pedigree. Okay, and what is the pedigree selection? It is the selection method which is used in segregating population of self pollinated species and keeps proper record of plants and progeny selected in each generation is known as the pedigree breeding. Okay, a selection method which is used in segregating population of self-pollinated species. Segregating populations means f1 and the further subsequent generations. We can say the f2 generation, f3 generation, f4 generations, and the further generations. Okay, it is the selection method which is used in segregating population of the self-pollinated species and keeps the proper record of plants and progeny selected in each generation is known as the pedigree breeding. Okay, in this breeding method, the individual plants with the desirable characters, individual plants from the f1 population, from f1 generation with the desirable characters are selected. Then these are threshed and harvested separately. Okay, the individual plants are selected from the f1 population on the basis of the desirable characters. Okay, then these are harvested separately, these are threshed separately, and their seeds are also collected separately. Okay, and these seeds, these are sown in the separate individual row in the next year to obtain the f2 generation. Okay, we are discussing how the f1 and the further segregating generations are handled in the self pollinated crop in the pedigree method. Okay, here the individual plants selected from the f1 population on the basis of desirable characters, these are threshed, these are harvested separately, their seeds are collected, these seeds are kept separately. Okay, then these seeds, of the F1 generation, these are sown in the separate individual rows in the next year to obtain the F2 generation. Okay, here in the F1 generation, the number of plants to be selected is completely depending upon the number of important characters involved, the importance of breeding program, and the assistance and facilities available. Okay, the number of plants selected, number of plants to be selected in the F1 generation, it is depending upon the number of important characters involved, the importance of breeding program, assistance, and the facilities are available. Okay, after the selection, these crops, these plants, these are threshed, harvested separately, their seeds are collected, and these are sown in the separate rows in the next year, in the next season, to raise the F2 generation. Okay, to raise the F2 generation. In F2 generation, what happens? In F2 generation, the different types of the plants appear. In F2 generation, different types of the plants appear which does not resemble with each other. Okay, and this F2 generation, it represents all the possible combination of the genes from the parental strain entering in this generation. Okay, I'm repeating this one. In F2 generation, the different types of the plants appears, and these plants, these crops does not resemble with each other, and these crops represent all the possible combination of the genes from the parental strain entering into the F2 generation. Okay, and therefore larger the F1 population grows, greater are the chances of obtaining the desirable gene combination in the F2 generation. Okay, therefore, larger the F1 population grows, greater are the chances of obtaining the desirable gene combinations. Okay, that means it is advised to select the large number of the plant from the F1 generation so that all the possible gene combinations plant breeder can get in the F2 generation. Okay, then in F2 generation, the disease resistance is also judged. Okay, disease resistance is also judged. Okay, that means the plants with the desirable characters and which are disease resistance, these are selected in this population. Okay, then the plants with the desirable characters and the disease resistance, these are saved, these are harvested, these are threshed separately, and the seeds of the each plant, these are kept separately, and these are then sown in the separate row in the next year to raise the F3 generation. Okay, desired plants, or we can say the plants with the desirable characters and the disease resistance from the F2 generations, these are harvested separately, these are threshed separately, their seeds are kept separately. Okay, and these seeds, these are sown in the separate individual rows to obtain the F3 generation. Okay, and that process, it is repeated up to the F5 generation. Okay, that procedure, it is repeated up to the F5 generation. But at the F6 generation, due to the continuous self pollination, these progenies will become homozygous and fairly uniform in their character. Okay, at F6 generation, the progenies at F6 generation, due to the continuous self pollination for the number of generations, these progeny will become homozygous and uniform in their characters. Okay, and the plants with the uniform characters, these are selected, these are harvested together, their seeds are bulked together to constitute the variety. Okay, remember that at the F6 generation, due to the continuous self pollination, the progenies will become homozygous and uniform in their character. Okay, and the plants with the uniform characters, these are harvested. Then these are harvested, these are threshed, and their seeds are collected, and these are mixed, these are bulked together to constitute the व्हरायटी. ठीक आहे, म्हणजे 6व्या जनरेशन मध्ये मात्र काय करायचंय, इंडिव्हिज्युअल प्लांटचं हार्वेस्टिंग करायचं नाहीये, तर एकसारखे कॅरेक्टर असणाऱ्या प्लांटचं सिलेक्शन करायचंय, त्यांचं हार्वेस्टिंग करून त्याचे सीड्स तुम्हाला मिक्स करून टाकायचे आणि ती एक व्हरायटी म्हणून तुम्हाला ती ट्रीट करायची आहे. Okay, at F6 generation, the plants with the uniform characters are harvested and bulked together to constitute the variety. ठीक आहे, पहिल्या जनरेशन पासून 6व्या जनरेशन पर्यंत, 5व्या जनरेशन पर्यंत काय करायचंय, तर इंडिव्हिज्युअल प्लांट सिलेक्शन करायचंय, इंडिव्हिज्युअल प्लांट विथ द डिझायरेबल कॅरेक्टर अँड हॅव्हिंग द डिसीज रेजिस्टन्स, आणि ते सेपरेटली हार्वेस्ट करायचे, त्याचे सीड सुद्धा सेपरेटली ठेवायचे आणि त्याचं सोईंग करायचंय पुढची जनरेशन ग्रोन करण्यासाठी. ओके, हे 5व्या जनरेशन पर्यंत रिपीट करायचंय, पण 6व्या जनरेशन मध्ये मात्र काय करायचंय, ते जे प्लांट्स ज्याच्यामध्ये सारखे कॅरेक्टर्स आहेत असे प्लांट सिलेक्ट करून त्यांचं हार्वेस्टिंग करून त्याचे सीड्स मिक्स करून एक व्हरायटी तयार करायची असते. Okay, the plants with the uniform characters, these are harvested and bulked together to constitute the variety. Okay, similarly, different varieties are produced and run into the comparative trials. Okay, similarly, different varieties are produced and these are run into the comparative trials. Comparative trial, or we can say the multi-location trials. Okay, these comparative trials, these are carried out in the different regional stations. Okay, and then their performance, then it is reported to the plant breeder, and plant breeder, one or two varieties are selected by the plant breeder. These seeds of the superior variety are multiplied and then these are distributed to the farmers. Okay, then after F6 generation, comparative yield trials are carried out at different regional stations. Their performance is reported to the plant breeder. Then plant breeder compare their performance, then select one or two varieties as a new variety, then the seeds of these superior varieties are multiplied and these seeds are then provided to the farmers for the commercial cultivation. Okay, that is the whole process of the pedigree method. Okay, the process, it takes about 10 to 30 years to release the new variety. Okay, pedigree method, it takes about 10 to 30 years to release a new variety in the self pollinated crop. Okay, that is all about the field technique which is used in the pedigree method. Okay, here in this photograph you can see the different steps which are involved in the pedigree method. Okay, here you can see the variety A, it is crossed with the variety B, and here the bulk plot, the here you can see the F1 generation. From the F1 generation, the individual plant selection, it is carried out, then the plants are harvested individually, their seeds are kept separately, then these are sown. Here in this one you can see the seeds, these are sown in the separate individual rows to obtain the F2 generation. Okay, these are space planted in the F2 generation. Just like that, the here also the selection is carried out in the F2 generations. Individual plant selection is carried out in F2 generation. Their seeds are kept separately. These are sown in the separate individual row to obtain the F3 generation. Okay, and this process, it is continue up to the F5 generation. This process, it is continue up to the F5 generation. But here in the F6 generation, to obtain the F6 generation, the generally the plants with the uniform characters are selected. These are harvested and their seeds are composited together to constitute a variety. Similarly, two or three different varieties are constituted, and then the comparative yield trials are carried out for the three generations, generally from the F8 to the F10 generation, and generally at the end of F10 or sometimes the F9 generation, the variety it is released as a new variety. Okay, the seeds of these variety, these are multiplied and these are then provided to the farmers for the commercial cultivation. Okay, this is the photograph, this is the figure which is representing the actual field technique which is used in the pedigree method. Okay, then the advantages and the limitations of the pedigree method. First of all, the advantages. The advantage is that this method it is well suited to the crops where the characters can be easily observed and individual plants can be easily harvested. Okay, it is useful, it is well suited to those crops where the characters to be combined in the cross can be easily seen, and these characters can be easily recognized, easily observed, and where the individual plants in a crop can be harvested conveniently. Okay, this is the first advantage of the pedigree method. Then the second advantage, it is the quickest method, as the new variety it can be selected and well tested by the time it reaches to the ninth generation after the cross. Okay, this method it is said to be the quickest method, though the it takes about 10 to 13 years to release the new variety. It is believed to be the quickest method, as the new variety can be selected and well tested by the time this variety reaches ninth generation after the cross. Okay, then the another advantage of the pedigree method is the plant breeder obtain the genetic information in this method which is not possible in the other methods. Okay, the genetic information which is obtained in the pedigree method it cannot be obtained in other methods like the bulk method or the backcross method or the multiple or composite cross. Okay, that's why this is the main advantage, principal advantage of this method. Okay, the genetic information which is obtained in the pedigree method is not possible to obtain in any other method. Okay, these are the advantages of the pedigree method. Okay, though there are certain advantages of the pedigree method, there is also a certain limitation to use this method practically. Okay, what is the limitation of this method? This method is most expensive. Okay, this method it is most expensive due to the consumption of more attention and labor for keeping the clear pedigree record of each selected plant separately in early generations. Okay, the pedigree record it is essential to maintain the pedigree record of each selected plant up to the F5 generations. For the 5 generation, it is very essential to keep the clear pedigree record of each and every selected plant. Okay, it requires, it is most expensive because due to the consumption of the more attention and labor for keeping the clear pedigree record of each selected plant separately in early generation. Okay, to keep the clear pedigree record it is very essential to give the more attention to each and every selected plant, and to give the more attention more labors will be required, and due to which this process it becomes more expensive. Okay, for the labors, the process will become expensive. Okay, because at the plant breeding stations, then another one important thing in this one is the at plant breeding stations, generally there a large number of the crosses are handled simultaneously. Okay, usually the large number of the crosses are handled simultaneously at the plant breeding stations, and therefore it becomes very difficult to pay and put so much attention and labor for keeping the clear pedigree records. Okay, as the large number of the crosses these are handled simultaneously on the plant breeding stations, it is very difficult to pay and put so much attention and labor for keeping the clear pedigree record. And that's why for this limitation, another method known as the bulk method is used to save the labor. Okay, and therefore the another method, bulk method, is proposed to use to release the new variety in the self-pollinated crop by using the hybridization method. Okay, that is all about the hybridization method. First hybridization method in the self-pollinated crop, and that is pedigree method. Okay, in the examination the long answer, 20 marks long answer question may be asked as give and detailed account on the hybridization methods in the self-pollinated crops. Okay, in this one you have to mention all the four methods, or the question may be asked as a short note in the examination separately as a pedigree method, or the bulk method, or the backcross method, or the multiple or the composite cross. Okay, this is the first method of hybridization in the self-pollinated crop, and the bulk method, it is the second method which is used in the self-pollinated crop. Okay, bulk method, it differs from the pedigree method. Okay, it is different from the pedigree method. Okay, it differs from the pedigree method. Okay, in this method hybridization is done between the selected parents with the desirable attributes. Okay, here the two parents having the different genetic constitution and having the desirable characters are selected, and these are crossed. Okay, the F1 generation seeds, these are space planted, and the seeds from these plants are harvested in the bulk. Okay, here individual plant selection is not carried out in F1 generation. Okay, here the plants with the desirable characters are selected, these are harvested together, and after harvesting their seeds are collected, and these are bulked together, or we can say these are mixed or composited together. Just like the pedigree method, here the individual plant selection is not carried out. Okay, this method is completely different from the bulk method. Okay, this bulking, it is done up to the F6 generation. Okay, that means the seeds which are bulked together in F1 generation, these are sown in the field to obtain the F2 generation. In the F2 generation, the plants with the desirable characters are selected, these are harvested together, then their seeds are collected together, these are bulked together, then these are used to raise the F3 generation. Okay, the same procedure is used up to the sixth generation. Okay, that means the plants with the desirable characters are selected, these are harvested together, their seeds are bulked together. Okay, and these seeds are then sown in the field to obtain the next generation. Okay, the same procedure it is repeated up to the sixth generation. Okay, but in F6 generation, the individual plants with the desirable characters are selected, these are harvested separately, and the seeds of the each plant are kept separately and sown in the individual rows. Okay, but at the F6 generation, the desirable individuals, these are selected and harvested separately, and the seeds of each plant, these are kept separately, and these are sown in the separate individual row to obtain the next generation. Okay, to obtain the next generation. Okay, then after the preliminary yield trials and the multi-location yield trials are carried out. Okay, after F6 generation, preliminary yield trials and multi-location yield trials. Multi-location yield trials, that means their trials, these are carried out at the different regional stations or the experimental stations. Okay, then their performance, it is reported to the plant breeders, and of these varieties, one or two varieties which show the superior performance, these are named, these are selected as a new variety. Then their seeds are multiplied, and these seeds are then distributed to the farmers for the commercial cultivation. Okay, the seeds, these are then distributed for the commercial cultivation. Okay, here in this photograph you can see the process of bulk breeding method. Okay, this method is opposite to the pedigree method. In the pedigree method, the individual plant selection is carried out, individual plants are harvested, their seeds are kept separately, these are sown in separate individual row to obtain the next generation up to the F6 generation. Okay, and after F6 generation, the plants with the uniform characters are selected and their seeds are bulked together. Okay, at F6 generation, the plants with the uniform characters are selected, these are harvested, and their seeds are bulked together, and after comparative trials, preliminary yield trials, the new variety is released. Okay, but here what happens in the bulk method? In the bulk method, the large number of the plants are selected, these are harvested together, their seeds are not kept separately, these are bulked together. Okay, and these are sown in the field to obtain the next generation. Okay, and this method, this procedure, it is repeated up to the F6 generation. Okay, but in the F6 generation, the plants with the desirable characters are selected. These are harvested separately, their seeds are kept separately, and these are sown in the separate individual rows. Okay, these are sown in the separate individual row to obtain the next generation. Okay, here you can see then the preliminary yield trials are carried out, and after preliminary yield trials, comparative yield trials are carried out, or comparative yield trials, we call it as a multi-location trials. These trials are carried out at the different regional stations, and after that, the by comparing their performance, the plant breeder will release one or two new varieties, and the seeds of these varieties are then multiplied, and these seeds are then provided to the farmers for the commercial cultivation. Okay, this is the actual field technique which is used in the bulk method. Okay, then the advantages of the bulk method, advantages of using the bulk method, this method is simple, convenient, and inexpensive. Okay, and less labor consuming, because in this one there is no need to keep the pedigree record. Okay, that's why it is inexpensive and less labor consuming. Okay, the advantage of the bulk method is it is simple, convenient, and inexpensive, and less labor consuming, because there is no need to keep the pedigree records in this method. Okay, then the limitation of this method is this method takes much longer time to develop the new variety. Okay, it takes more time when compared with the pedigree method to release the new variety. Okay, but it minimizes the labor as the plants are not paid any individual attention, and sometimes the plant breeder has no option except to use this method for the development of the new variety in the self-pollinated crop. Okay, sometimes the plant breeder has no option except to follow this method. Okay, that is all about the bulk method which is used in the self-pollinated crop. Okay, then here you can see, do not get confused between the pedigree method and the bulk method. That's why here in this slide I have mentioned the certain differences between the pedigree method and the bulk method. Okay, here you can see in the pedigree method the individual plants are selected and these are bulked together at the end. Okay, these are bulked together at the F6 generation. But the bulk method is different. Here the selected plants are bulked and the individuals are tested separately at the end. That is, individuals, these are tested separately at the F6 generation. Okay, exactly opposite to the pedigree method. Okay, in the pedigree method individual plants are selected and these are bulked together at the end. Okay, and here the selected plants are bulked and individuals are tested separately at the end. Okay, then in the pedigree method less time is required in producing the variety, while in bulk method it is time consuming. It will take more time in producing the new variety. Okay, in bulk method more time is required in producing the new variety. Then in pedigree method, as the in early generation it is essential to keep the record of each and every progeny of the selected plant. Okay, that's why this method it is tedious and complicated. But in the bulk method there is no need to mention, no need to keep the pedigree record. That's why the bulk method it is simple and convenient. Okay, in the pedigree method it require more attention, it will require the more labors to keep the pedigree record, and that's why this method it becomes most expensive method. Okay, but in the bulk method it is inexpensive. Okay, it is inexpensive because it will not, it will does not require more labors. Okay, and that's why the bulk method is inexpensive. Okay, these are the differences between the pedigree method and the bulk method. Okay, the two methods are remaining. The third one is the backcross method and the fourth one is the multiple or the composite cross. Okay, we will discuss the backcross method and the multiple or composite cross in the tomorrow lecture. Okay.